Good morning, everyone. Welcome to today's session of Indian webinar series, Introduction to Computational Drug Design. It is co-organized by Schrodinger and PCI. I would like to take a moment to thank our collaborators and the panelists from JSS and Manipal, and also the IT team of JSS for hosting these sessions. Today we have one session about computational formulation design by Dr. Sudarshan. So during this course of the presentation, if you have any questions, feel free to drop them in the chat section of the YouTube link. We will have a Q&A session at the end of this talk. So without further ado, I would like to welcome our only speaker for today, Dr. Sudarshan.
Good morning, everyone. Sylvia, am I loud and clear? And
Yes.
the slides are visible, right? Yeah.
Yes.
Okay. So, good morning, everyone. So yesterday, I gave you a brief introduction about how quantum mechanics can be used for drug discovery purpose, computational drug discovery. And today we are going to look at computer-aided formulation design or pharmaceutical formulation. So how we can use computational tools or molecular simulations to understand pharmaceutical formulation problems and address a few of the issues that are bottlenecks in the formulation design. Okay, so if you look at the pharma market in India, so India is becoming the pharmacy of the world. So that's a true statement, actually. So if you look at even the corona situation, people are trying to come to India and do their synthesis or do their product development in Indian using Indian pharma companies. For example, Russia is trying to collaborate with the Dr. Reddy's for developing their vaccine for coronavirus. So like this, we are getting a lot of business from foreign countries or Western countries and trying to produce pharmaceutical ingredients in India and also pharmaceutical formulations, the entire generic medicines. So we do a lot of export from India. If you look at the business in India, it's growing in the pharma industry, and pharma industry is one of the main pillars of Indian economy also. So it's doing really good. And most of our exports go to North America and some of them to Europe, and to Africa also we send our generic medicines. So it's a growing industry where we develop formulations, or we develop we synthesize molecules for the Western pharma companies. We give it to them to their need. And but when you look at the number of drug molecules that came out of India, it's actually none. We develop molecules to a level where these are becoming a lead molecules or becoming an IND candidate. So then we pass it on to big pharma companies simply for this reason. So cost to develop new pharmaceutical drug molecule is beyond $2.5 billion. $2.5 billion. It's a B. Okay, so this was already in 2014. You can see it's November 2014. But it's in 6 years time the cost has further increased, maybe because of the computational tools. The cost is kind of now reducing, but overall still it's in the billions. When we speak about $1 billion, so that's not possible for an Indian pharma company to spend and take it to the market all the way possible. So that's why we develop to a level where we can get a molecule into a FDA's approval for trial runs. So the moment it hits the clinical trials, we collaborate with Western pharma, or we completely license the molecules to Western pharma companies. So that's what is happening. That's how Indian pharma companies make their money from the new drug discovery research in India. Okay, but then where do we get all our money from? As I told you, the business is growing. The business is really good. Where do we get the money from? It's generic drugs. The generic drug is the ones which came out of the patent rights, so anybody can generate it. But when we speak about generic drugs, it's not only necessarily the developed company, the company which developed that particular drug takes the patent on only the API molecule. So API is one part of the patent. They also take the patent on the formulation. They also take the patent on the process and methodology. Usually this process on methodology and the formulation, they take it a bit later, so that they can have a complete control of this drug molecule for slightly longer years. Okay, so that's why when some API molecule is coming out of the patent, people try to work on that particular drug molecule for a new formulation and also for the new process and methodology, so that we can come up with completely new set of product for a cheaper price. As you can see, for the generic drugs, you can see US is very expensive, India is kind of probably the cheapest. So from here we can export, and then we can make money by exporting generic drugs. If you look at the US pharmacies, about 16% of the drugs are coming from Indian pharma companies like Sun, Zydus, Lupin, Dr. Reddy's. So this is happening because we are able to generate right kind of formulations, right kind of process and methodology to break the patent limitations for the drug molecules which are coming out of the their patent period, so that come up with a completely new set of product and export it to Western countries. So this is what exactly we do to make more amount of money in India. Because by selling the molecule or licensing the molecule, newly developed molecule, small molecule, we are getting only a small amount of money compared to what the whole business is doing. But the major amount of money is coming from this particular development, exporting generic drugs. Okay, so when we speak about the pharmaceutical formulation development, it could be any different type of formulation, from ranging from tablets, capsule, liquid orals, or even to the topicals and syrup, um, effervescence. So it could be multiple different formulations. What we are trying to understand here is the molecular level interactions. We are trying to understand, zoom it in to the molecular level and try to see how things are happening at the molecular level in the given formulation, why particular thing is working and particular thing is not working. That's what exactly we are trying to find out using the molecular simulations. So when we speak about formulation development, usually they come up with some kind of problems. Why? Because when we speak about drug discovery is a holy grail problem. So we are trying to find out one molecule potentially that can bind to the given target structure, right? So that's only the API molecule. The API molecule designing is now becoming more and more simpler problem because of the computational tools. Thanks to the modern day computational tools, we are able to quickly solve the problem of API binding to the protein structure. And also even ADME properties are now very well parameterized thanks to tools like FEP plus, and so that we can have multiple molecules in the pipeline. So the one molecule that can pass through all the filters and then come up with a lead molecule or IND molecule. So that part is kind of very well stabilized now. But when it comes to formulation, that's a becoming more and more issue, because the modern day drugs, especially for the cancer treatment or any other modern day complex disease treatment, the drug molecules are potentially insoluble in water. So we have to find out from the solvent to all other components of the formulation, which might contain 3 to 20 different components depending upon the complexity of the formulation. It's a huge melange. So but when it's a melange, there are potential chances of one interacting with the other, potential chances of chemical reactions. So when there are chemical reactions between them, that could potentially lead to impurities. Then the impurity profiling becomes even more complex problem, because we don't know which one is reacting with which one, what kind of impurities coming in, and what is the timeline for this impurity to come out, and whether it's going to form a manageable or a completely uncontrollable quantity of the impurity. So then how do we address this problem? So these are the problems which formulation scientists face. And when you look at the cycle of taking the molecule into clinic, or taking the molecule into market. So the discovery happens at the start, as I told you, the API discovery. Then the product characterization, that's where we discuss about what kind of way we are going to deliver this API into the body, whether it's going to be a liquid dosage, or it's going to be the solid dosage, or it's going to be what kind of form and how much of dosage is needed and stuff like that. Then the formulation and delivery system has to be defined. So if that goes wrong, we have to sometimes go all the way back to discovery and come up with some kind of modification, which is again, if you go further down the line, if the no cost incurred is going to be increasing all the time. If you fail at that particular stage, if you want to come back to the original state and start the process again, it's all huge loss of money, time, and manpower, no skilled manpower. To avoid that one, what companies do is nowadays they start as early as during the discovery process, work on the pre-formulation studies. So when they do the pre-formulation studies, they don't want to spend huge amount of money, or the huge amount of uh, resources to do these pre-formulation studies, can we use some kind of simple simulation tools, some kind of simple understanding of the materials at the molecular level, so that we can have something which is kind of already ready when the stage when we come to the stage of formulation, so we can use this information for effective completion of formulation development and also understanding the delivery process. So that's where molecular simulations play a crucial role. And I'm going to show you a few examples, few case studies, the typical problems in the pharmaceutical formulations, and how we can address these problems using Schrodinger's material science suite. So that's what I'm going to show you. So as I told you, material science suite, which the word material science means like basically anything that has mass, anything that occupies space is a material. So we can apply to any different kind of industry, from organic electronics to alloys and crystals. So today we are going to speak about the pharma formulations and delivery. Okay, so it has different applications in different industry. Today our focus is on pharma formulations and delivery. Okay, so here are the few problem sets. Choosing the excipients. So let's start a completely new formulation project. So we have a API molecule in hand. For the API molecule in the hand, we have to select, we know what kind of dosage form we are going to build. So we have to select the excipients. So how do we select the effective excipients? Using computational tools. So that would be one of the problems. So I'm going to show you an example in that, like how do we choose excipients using computer simulations. So next, understanding the role of the excipient. So for example, there is a formulation. We don't know this formulation is working for some reason, but why it is working, we can't give an explanation. What is the role of each and every component in this formulation? We still don't know. So there are 6 components, but 7 components. Why these components are coming together and making this formulation work? So we want to understand what is the role of each and every component. So once we understand the role of each and every component, we can find a way to replace the expensive component with a cheaper component. Okay, that would be an ideal way. And also, if there is a commercially available formulation, so we can pick up that formulation, try to understand what is the role of each and every component, so that we can replace the potentially patent bound components with a completely new component, so that we can come up with a completely new formulation also. Okay, then the third problem would be the physical and chemical stability of the formulation, which is kind of most common problem. Out of hundreds of formulation which we develop, only 1 or 2 of them are going to be successful. Why the others are failing? What is the reason begin failing? So we are expecting a complete clear solution. Instead we are getting a turbid solution or a precipitate, or we are getting a nice spherical nanoparticulates. Instead we are getting a flakes, and we are getting sticky mass over single blob of entire component. So that kind of situations are quite common in the formulation development. So how do we address this kind of physical stability issues, and also chemical stability issues? When it comes to chemical stability, so as I said, anything can react with anything inside the formulation, so we can get impurities. So how do we get this reactions blocked, or how do we understand these reactions in an effective way? So these are the things which we have to understand when it comes to physical and chemical stability of the formulation. And impurity profiling, so that is where we know that when the chemical instability is happening, we have a lot of impurities that can come out. So how do you know these impurities come out, and in what concentration they can come out, and how long it would take for them to accumulate in the noticeable quantity? So these are the problems. But before we go to that part, we have to understand what kind of impurities can come out of this formulation. Okay, whether these impurities are going to be hazardous or not. So that has to be understood. That's called impurity profiling. So which can be done using computational tools also. Then understanding the shelf life of the formulation. So how long this formulation would be stable, because that would eventually give us an idea of what could potentially be the expiry time limit of the given formulation. So these are the usual problems which are common in the formulation development. It's not only restricted to these problems. There are quite a few other problems, like coming out of the patent by understanding the process, so that we can completely come up with a new formulation. So those kind of things are also happening in the industry, and understanding the synthesis of even the excipients and stuff like that. But again, those are the projects which we have done for pharma companies, which are now NDA bound. I can't share those kind of examples, but these are the examples which I can share with you today. That's why I selected only these topics, but it's not only restricted to these topics. These are the few example topics in the area of formulation development. It's not only restricted to only these problems. If you have particular research problem in the formulation development, we can definitely look into that one, and then try to see what kind of simulations we can run, and what kind of properties can be understood, or what kind of designing can be done at the molecular level using computer simulations. Okay. Yeah, so here is a first example, choosing excipients for formulation. So we have this particular script called disorder system builder, which would allow us to build multiple disorder systems, that means amorphous systems of the given components. So once we have this given components, we can calculate their cohesive energy. So cohesive energy is the amount of interaction energy that's there between the molecules. So that cohesive energy can be used to calculate something called solubility parameter. Hildebrand solubility parameter, Hansen solubility parameter. There are two different types of solubility parameters that are there. We can calculate both of them. So once we calculate this Hildebrand and Hansen solubility parameters, so these parameters can give us whether particular components are going to be miscible with each other or not. When you are putting them together, they are going to be happily interacting with one another, or they're going to phase separate from one another. When you are putting them inside a particular solvent, whether these two components are still going to be interacting inside the solvent, or they're going to completely go away from one another. So this is the important part to understand, right? When we are developing a formulation, we have a drug molecule, we want to select a drug carrier or enhancer or any other component that has to have a favorable interaction with the drug molecule so that they stick together throughout the entire process of formulation development. So to understand that one, we have to understand the miscibility between these two components. It's not necessarily in the solution form. It could even be a powder or in a solid form. So how they are going to interact with one another. When we mix them, how they're going to be happily mixed together. So that's the thing which we have to understand. That particular component can be understood from solubility parameter. Okay, so the thumb rule here is the solubility parameter difference is less than 7. That means they're going to be miscible. If it is greater than 7, they are not going to be miscible. There are quite a few publications out there which explain this particular part. Okay, so we can really understand that one. In this case, we have ibuprofen as our drug molecule. These are the drug carriers: PVP, luteol, F68, maltose, sorbitol, and xylitol. So these are the drug carrier molecules which we want to try out with the ibuprofen. So these are the things which are coming from the experimental values. You can see 20.9 is the solubility parameter for the ibuprofen. 22.5 and 19, this particular column is the value for the carrier molecules. So this experiment usually takes days together to calculate this solubility parameters. For 6 different components, it would take days or maybe more than a week for the entire table to be populated. But whereas when we have simulations, this entire thing can be done within a day, okay, because we have the GPU accelerated MD engine. This is done using MD simulations. We can do within few hours time, we can populate this particular table. And then the difference between them is given in this particular two columns, delta tau. I can see the simulations are predicting as close as to the experiments. So basically, we can say whether two components are going to be miscible or not in a given formulation. That would allow us to pick the right kind of excipients for the given formulation. So this is one of the approach through which we can pick the excipients for the given formulation. Okay, so we have done the similar kind of study and published a paper in year 2018. So here in the first movie, you have two different components. Okay, so the cyan one and then the pink one, these are the two different drug molecules which are kind of having different characteristics. They are not soluble in the same solvent, so they are supposed to be soluble in a mixture of solvents. So with the solubility parameter values, we were able to select a mixture of solvents where both the molecules can be happy. And you can see the first movie, they are all happy. They are happily floating around. All the drug molecules are happily floating around in a mixture of two different solvents, red and green. So they are happily floating around. It's like in the beaker, you have four components: solvent 1, solvent 2, drug 1, and drug 2. So they are all happy. And then the second component, the second movie, you have second beaker where you have a big drug carrier molecule, a big polymer chain, PEG 4000, PEG 2000, PEG 20,000, or luteol. So you can take any different type of drug carrier molecule. So that's floating inside a anti-solvent, a solvent where these molecules are not having favorable interaction. So then you pour the beaker 1 into beaker 2. So when you add the beaker 1 to beaker 2, you have this incoming drug molecules which are now having unfavorable interactions outside. So they don't want to go outside. They have to come aggregate together and form a closed loop with each other. They are aggregating because they like to interact with each other compared to the anti-solvent molecules. Where do they find asylum? They find asylum inside this big drug carrier molecule. Then in the third case, where we evaporate all the solvent molecules, only the anti-solvent molecules are present, because the anti-solvent molecules are only present. Now I don't want to go out at all out of this nanoparticulate. So then the molecules getting trapped inside this particulate So then we get a nanoparticulates out. So this is how. So this is a three component system. In this case, we have drug carrier and drug one and drug two. We can add further components like surfactants and other excipients, so that we can see the nice formulation getting formed during the course of the simulation. So this is how the nanoparticulate is getting formed at the molecular level inside any beaker or any experiment. So we can basically trace them back to the molecular level and explain why particular thing is happening. In this case, it's working. In suppose if it is not working, why it is not working, we can explain that also. Why it is not working using the simulations. Okay. So how do we do that one? What do I need to do this kind of simulations? So to do these simulations, what do we need is the chemical structures and their composition. Okay, let me come back to this slide. So chemical structures and composition. In this particular formulation, you can see, so these are the chemical names and their composition. Okay, once we have these chemical structures and chemical composition, okay, so we can convert them into number of molecules. Once we convert them into number of molecules, then we can build up a simulation box containing particular composition of all the components of the formulation. Once we have all the components of the formulation inside the simulation box, we can run it for any number of length and then try to visualize what is happening at the molecular level. Okay, when you go to more and more components, it becomes more and more complex. But when we have only few components, we definitely can understand what kind of interactions can happen at the molecular level between one component with the other component. Okay, we can study the effect of concentration, effect of temperature, and also what is the role of each and every component. What happens when we completely remove that component? What happens when you replace that particular component with a excipient with some other completely new excipient? So these things would allow us to have better understanding of what is happening at the molecular level. Okay, so for that we use this particular builder called disordered system builder, where we can add as many as components we want. Once we have as many as components we want in the right stoichiometric ratio, we can set up the percentage of each and every component. Once we have the right kind of components added, we can build up a simulation box containing all these components. Then we can run MD simulation. MD multi-stage workflow would allow us to run the MD simulation. Completely equilibrated system can be built. From the completely equilibrated system, we can really understand the interactions between the components by calculating the radial distribution function or any other property of interest, hydrogen bonding interactions between different components and stuff like that. Okay, so that's how it is done using computer simulations. Okay, so there are other examples like chemical and physical stability. In this particular example, we have a drug molecule and then we have a sweetener molecule. Okay, so they are supposed to be soluble. They are supposed to be happily interacting to one another in the solvent. Rather, they are forming a thin layer of sticky mass precipitate at the bottom of the beaker. So what is happening? So experimentally, if we consider this problem, we have to understand why this sticky mass is coming. So it could come from the impurities. It could come from the interaction between these two components. There could be chemical reaction there. If there is a chemical reaction, what kind of chemical reaction is there? So how do we get the structure of this impurity, and how long it would take it to form this kind of impurity? So it's a kind of quite a lot of questions are there. Quite a lot of experiments has to be done to understand this problem. Whereas in simulations, we know that this is forming a precipitate. Why it is forming a precipitate? What is that in the precipitate? That's the question we want to understand. Okay, so we did a solubility parameter analysis. As per the solubility parameter analysis, they are supposed to be soluble and they are supposed to form a uniform solution. Okay, so you can see, so they are within the range. So all the three components are supposed to be soluble. They supposed to form a uniform solution. But we know for sure. Okay. Yeah, we know for sure, they are forming some kind of precipitate. Why this precipitate is coming into picture and how it is happening. So we looked at what is the potential reactive site inside this given drug molecule. These two particular bonds are potentially weak. That's their pKa values. That means they're going to take up a hydrogen. So these potential bonds can break into pieces. So then when we take this molecule and do a substitution reaction, so when you substitute this particular fragment with an incoming fragment, you get a product A. When you substitute this second half with this particular fragment, you get a product B. So you have product A and product B, which can potentially come out of these reactions. Okay, substitution reaction. When you take this substitution reaction and try to understand whether the reaction is possible or not, yeah, they are endothermic reactions. Maybe under given conditions, maybe under the given chemical environment, it might happen in a small quantity, but not in a complete way. But that's why we are getting a thin layer of precipitate at the bottom of the beaker. But the reaction can happen. The product A can definitely form. Product B on the other hand, it's not going to form at all, because the energy required to make this reaction is really, really huge. That's why product B is never going to form. Product A is definitely going to form. Okay, so if the product A is going to form, okay, so if I put these two components together, a small amount of product A is going to form. We can give a computational IR spectra of the product A. Okay, so that we can take a AT IR spectra of the impurity or the precipitate, and then compare its spectra with the computational IR spectra, whether is this the component that is happening at the bottom of the beaker. And also we went ahead and did a simulation of that impurity dispersed randomly that in the simulation box, water box, and then calculated how long it takes for it to aggregate and what is its viscosity. You can see it's aggregating, it's phase separating from the solution environment. It's kind of phase separating. You can see it's all aggregated together. Why? Because its solubility parameter is 22 compared to 48 of water, because of which no, it's not going to like water. It's going to completely phase separate. That's why it's reaching the bottom of the beaker. And viscosity of this one is 480 centipascals compared to 1 centipascal of water. That means it's sticky. Okay, 480 centipascal is the viscosity of engine oil. So it's obviously going to be sticky. So that's why it's forming at the bottom of the beaker. So this is all been done in 4 days time. Okay, in 4 days time, we could be able to provide what is happening, a molecular level explanation of what is happening, why the precipitate is forming at the bottom of the beaker. Experimentally, it would have taken weeks together to find out what is the cause of this problem. But in simulations, we were able to explain why this particular thing is happening at the molecular level. Okay, and here's another example of impurity profiling, metformin synthesis. So A plus B is reacting, and then C1 plus C2 product are coming out. C1 is the major product, that's metformin. So that particular which is used for treating high sugar levels. Okay, so here you have major product, you have small minor product, which we want to find out what is the impurity, the minor product which is coming out of this reaction. Okay, so we have this beautiful panel called reaction channel enumerator. You give the A plus B structure and then ask it to build all the possible association products. They will build all the association products. If you want to find out AB, which is breaking into pieces, A plus B, the dissociation products also can be calculated. Same with the single replacement, double replacement, all these reactions can be done, and all the possible combinations can be plotted out. In this case, if you look at it, we have 1, 2, 3, 4, 5, 6 different sites are there. In this case, it's a symmetric molecule, so we have only 2 sites are there. So that's a 6 factorial into 2 factorial. We are getting about 1,440 products that can possibly be built from these 2 molecules. Okay, but then in that case, we have to run 1,440 simulations to find out which could potentially be the C2 structure. Okay, by different combinations, permutations and combinations. But if you can get any information about the impurity, that would allow us to reduce the number of simulations that we have to run. Okay, if we say mass, we can set up a mass criteria. We can set up the impurities which are forming with certain mass. Only above that mass we are going to consider. So we can remove all the other unwanted products. If I have a fragment detail, let's say I have particular fragment in my group. So let's say like I have CH3 and H2 in my group. CH, so then in that case I can search for molecules with only such kind of fragments, and then ignore rest of the molecules. When I have functional groups, when I have a primary amine, when I have a secondary amine in my group, so then we can use that particular functional group to screen the molecules, 1,440 products, and then come up with only molecules containing that functional group. Or the scaffold, I have a 6-membered ring, I have a 5-membered ring, I have a 5-membered ring fused to 6-membered ring. So this kind of scaffold information would allow us to reduce the number of simulation that we have to run also. In this particular case, we know that C2 and C1 are having the same mass. That's why they are finding it difficult to separate these components from each other. Okay, so C2 is having the same mass as C1. So when we look at it, so these are the 6 components with the same mass, and here is our metformin. So that has this mass, and all the other components which are having the same mass. Okay, so when we take all these components, go to this reaction workflow, which is again quantum-based workflow, quantum mechanics-based workflow. You can add your reactants, you can add your product structure, and then calculate your delta H and delta G of the reaction. And delta H and delta G of the reaction would give us whether the reaction is going to be spontaneous or non-spontaneous. Okay, so if you look at it, C1, which is our major product metformin, which is an exothermic reaction. When you look at all the other reactions, they are all endothermic reactions. Okay, so whereas only one other component, which is an exothermic reaction. So these two components are exothermic reactions. That means potentially these two can form. So C1, which is going to be obviously the major product, C6, which is going to be a minor product. Okay, so this is our impurity, and this is the structure of the impurity. Again, this is done within a week's time. What is the impurity structure and how this impurity can be, profiled and understood. So impurity profiling, an example for formulation development. Okay, so this slide which I showed you yesterday also, understanding everything about the API molecule. So when I have the API molecule, if I have the API molecule, if I want to understand what is the most reactive site in this API molecule, so that can be understood using Fouquet functions. We can say where the acrophile is going to attack in this molecule, where the nucleophile is going to attack in this molecule, and then find the weakest bond in the molecule. So that can be done using the bond and ligand dissociation panel. With this panel, we were able to find out what is the weakest hydrogen as well as what is the weakest bond in the molecule, so that that bond can be broken into pieces. What kind of impurities can come out of this molecule? And then the third, so when I put this molecule under different pH conditions, so how this pH is going to impact the molecule, how it's going to behave under different pH conditions, that can be understood using the pKa values. So 3 different panels would allow us to have a better understanding of our API molecule. Eventually, this would allow us to have a better understanding of the formulation. It's not only for the API we have to do this one, we can do it for the small excipients also. So we can take the excipients, we can see how these excipients are going to behave under different conditions. So that can be understood also. Okay, so and here is another example of a beta cyclodextrin, which is used as a taste masker in most of the cases. So beyond a certain critical concentration, the beta cyclodextrin is going to aggregate, whereas below certain concentration, it's not going to aggregate. In the first movie here, you see that it's above the particular concentration, you can see the aggregation is happening here. For the first 200 nanosecond, it was not happening, but the second 200 to 1200 nanosecond, you can see the aggregation is happening. It's forming a nice aggregate, okay, above the critical concentration. So here is the critical concentration. Anything above this concentration, it's going to aggregate here like this. The aggregate size you can see. So why it is aggregating and how it is aggregating, that can be visualized. Okay, so below certain concentration, you can see below this concentration, we have the simulation. Okay, 6.7 mg per ml. So here you have 13.4 mg per ml. So in this case, it's aggregating. In this case, it's not aggregating. So we can visualize such kind of phenomena also. So what is happening? What kind of interactions are driving this kind of aggregation process, and why the aggregation is going to be happening for the given excipient system or given component system. So visualizing and providing molecular level detail of why it is happening. Okay. Yeah, and yeah, the drug, the how to say, the liposomes. So where we want to see liposome kind of paste formulations. This is coarse grain simulation. In the coarse grain simulations, when we have a drug loading, so when you have different types of drug loading, how these drugs are combining together and forming lamellase, or what kind of interactions are happening between the liposome walls with the drug molecules, concentration of the drug molecules. So how this penetration is going to happen, that can be simulated using coarse grain simulations again. Okay, so we can understand why this drug penetration is going to happen this much time, what kind of interactions are there. Once it makes the hole, how it's going to penetrate, all those things can be visualized using coarse grain level simulations. So this is another kind of example for liposomes, which are used as a drug carrier material. Okay, as I told you, okay, understanding the pharmaceutical formulations from the molecular context is key to avoid increased cost of failure at the later stage. So we have to really spend a lot of time in the pre-formulation studies to pick right kind of excipients. Using simulations, we can pick right kind of excipients and understand the problem effectively, so that we can come up with the right kind of combinations for the experiments which we want to run for the formulation studies. And also, if the experiment is failing in for any unexpected reason, we can trace it back and try to find out why the experiment is failing, how we can address that failure, and what are the components that are making that failure happen. So how do we replace that component with a much better component? So that can be understood using computer simulations. These are the few examples which I want to show. There are quite a few other examples like chiral separation. So understanding chiral separation using molecular simulations, which is kind of an area of interest. And then choosing coformer selection. I have an API molecule which is insoluble in water. So then how do I find out a coformer that can potentially have behave like a single molecular unit when they are interacting with one another? and these interactions, how do they play a crucial role in forming the coformers, and what is the choice of coformer which I can pick from the list of coformers that are available for the given API molecule. So those also kind of those are problems which we are currently addressing. Currently, we are working on, but there are quite a few other problems also which I can't discuss openly. But these are the few examples which I want to show you, which I wanted to show you. So with these examples, I will stop my presentation. I'd be happy to answer any of your questions if you have any. Okay, I thank my team for this wonderful opportunity, and they did a lot of effort in providing you with a lot of good content. Let's wait for the questions now. Over to you, Sylvia.
Thank you, Dr. Sudarshan. So we have received a few questions. Just a reminder to everyone, if you have any question related to this presentation, please add them to the chat section of YouTube so that we can ask our speaker. So the first question that I received is, sir, while explaining simulation to understand the role of excipients, you mentioned you mentioned about a composition of drug, the paclitaxel 100 mg. So on what basis this composition was decided?
Well, the composition is that's coming from how to say, the studies. Like, if you want to build a formulation for a given component in a given concentration, so that's the concentration which you want to take, right? Like, basically, I want this particular drug molecule is required in this quantity. So how do we know this quantity is required? We know from the binding affinity, we know from the IC50 value how much of active quantity is required to make some effect on the given physiological system, right? So to that IC50 value can be converted into the concentration. That concentration can be converted into the amount of drug molecule, amount of API molecule that we have to send inside the body in a single dosage. So once we know in a single dosage, we need to have this much of quantity of API, then around which we can build our excipient concentration. So it's coming from actually the IC50 values. So the amount of concentration required to make some impact inside the body. So that decides what kind of concentration is required for any dosage form. Yeah.
Right. So you talked about the excipients and the API. So there's just another question. Just like, how can you decide the composition of all the components?
Yeah.
Yeah.
As I told you, like one by one, we can pick up the right kind of excipients. For example, I had decided my API. Okay, so API is for certain, that's the one which I'm going to use. Then next I have to select the drug carrier. So when I want to select a drug carrier, the drug carrier and the API has to have a favorable interaction between them. So when I put them inside a solvent, when I put them inside the body, they are supposed to have an interaction till the point it reaches inside the site of interaction or closer to the site of interaction. So that means they are supposed to be miscible with each other. So we can use the solubility parameter to select the drug carrier molecule. The same way, I want to select the enhancer molecule. The enhancer molecule is to go with the API molecule till the point where it reaches the cell membrane, where it would help it to penetrate to the cell membrane. So I want to pick up the excipient molecule, which is going to have a favorable interaction with my API molecule. So then I have to find out the solubility parameter of the enhancers, find the one which is going to be miscible with my API molecule. Whereas in the surfactant case, I want the surfactant to go only to a certain extent. So I don't want surfactant to have a favorable interaction with the API molecule. In that case, I want to find out a molecule where they are not going to be miscible, but the surfactant is going to be there in the formulation to make that it goes through the system. But once it goes through the system, the API molecule should not have any interaction with the surfactant. Then I have to find out a molecule which is having a solubility parameter difference of greater than 7. So we can play around, either it's going to have a favorable interaction, it's not going to have a favorable interaction. So how these components can be picked up? So we can pick up one by one. So at the end of it, we will have complete formulation from these simulations, and which we can simulate again to validate what kind of interactions are happening at the molecular level. Is there a chemical reaction between them? If there is a chemical reaction between them, what kind of impurities can come out of them? And how do we profile these impurities? So even up to that extent, we can use simulations to understand that.
Thank you, Dr. Sudarshan, for that explanation. The next question is, what is the meaning of anti-solvent?
Anti-solvent is the solvent where the compounds are insoluble.
Right. So the next question is, can we also use this software to model and simulate the dissociation and drug bolus release from nanoparticle carriers? Maybe something like dextran.
Yep, we can do that one. So basically, yeah. So what we want to understand is like the dissociation energy of the drug with the nanoparticulates. So we can definitely do that one. So we can calculate what is the binding energy between this nanoparticulate and nanoparticle and the drug molecule. So we can calculate their together energy and the individual energies. From there we can calculate what is the binding energy or amount of energy that is required to break this bond, and under which conditions this bond can break. So that can be understood. Yes.
What will be important consideration for developing topical formulation?
Well, topical formulations, again you need a skin permeation. We have models for skin membranes. So again, that's one of the example which we worked on. Also we have skin membrane models, and when we put the topicals on top of it, it has to have some kind of penetration through the skin membranes, because it's not only on top, it goes into few nanometers inside the skin. So when it penetrates through the skin, how long it would take for it to penetrate, and what kind of penetrant enhancers are required to facilitate the permeation, and if they are permeating, and know which molecules it's having, which skin type of molecules it's having favorable interaction, because skin is made up of plenty of types of ceramides. It has some cholesterols, it has some fatty acids. So what kind of interactions are there, and how the penetration is happening, so that has to be understood. So for that, we can do simulations also, because we have skin membrane models through which we can try to understand how the topical permeations are going to be happening at the top of the skin.
Thank you for that answer. Moving on to the next question. What interactions are important or can be probed using Schrodinger for drug molecule and polymer explicitly?
Well, we can understand everything about this drug and polymer interactions. Basically, when you put a drug and polymer together, so whether they're going to be together, first of all, they're going to be together or not, that has to be understood. Second, what kind of non-bonded interactions that are going to be there between these molecules. Basically, like you do the docking with the protein, but here it's not docking. It's simple, straightforward MD simulation, and we can calculate what kind of no non-bonded interactions are there between the polymer and the ligand molecules. What happens when we change the concentration? What happens if we change the polymer chain length? So all those things can be understood. Yeah, anything and everything that is there at the molecular level can be understood using polymer excipient interactions.
Right. Now we have a very specific question. What will be the choices of proteins such as antibody for topical delivery to target local injury?
Well, to be honest, I have no answer for this particular question. Well, it's you have to do the literature search. I don't have an answer for this one.
Right. So just move on to the next question. The next question is, sir, in impurity profiling, if the impurities are more, then how do we conclude?
Well, if the impurities are more, that means that particular formulation is not going to work. First thing, and that's a clear, and that's a clear answer. And why these impurities are coming? So where these impurities are coming from? So that has to be understood, right? So the impurities are either coming from the source itself, then we have to change the source. If the impurities are coming from some kind of chemical reaction between the components, so can we replace this one of this component, so that we can avoid this impurity formation? So that's exactly what is happening. So in the pharma industry, what they use is they use ultra high purity components. So there is no chance of the impurity coming from the source. So when then the impurities are coming, it's mostly because of the chemical reaction between the components. So if the chemical reaction between the components is possible, then we have to find a way to block them. So how do we block them? Basically, replace one of the component with a other component where the reaction energy barriers are so high, so that the impurity formation is going to happen in a very long time. For example, if you take H2 plus O2 forming H2O, it is an exothermic reaction. Eventually, it should happen, but the energy barrier required for that reaction to happen is so huge. So in a million year, you can find one H2 or O2 molecule reacting together, forming H2O molecule. The same way, we can find two components which are potentially reactable, but their energy barrier is so huge, there is no chemical reaction possible in the meaningful time period. So that kind of component can be picked up from the use of simulations.
Thank you for the question. Right. The next question is, can we use cell penetrating peptides as drug delivery system, and using Schrodinger, can we understand the stability?
Yes, of course, you can. Yeah, you can. There are different cases, different things which are used as cell permeating enhancers or permeating systems. So people use nanoparticles, people use, even small peptides or liposomes and stuff like that. But again, definitely a peptide can be used. A peptide can be tagged along with the API molecule so that it can drag it inside the cell. So yeah, that definitely can be simulated and understood what kind of mechanism is happening for the penetration. Yes.
Right. So there is one more question. Can we model a protein topical formulation with Schrodinger?
Well, I need more details. The topical formulations, as I said, we have membrane models for the skin. We can definitely put anything on top of this membrane models and try to see the interaction of the components with the skin membrane. But I need more details to give further information on that one.
Thank you. That's it. I think I have covered all the questions for this.
Hi,
Yeah.
Sylvia. Sorry to
Yeah.
interrupt.
Sorry.
Hi, Sudarshan Kaushik here.
Yeah,
I have a question for you. Can you go to slide 11
11.
or probably 11 or 12? I don't know which one exactly. Sorry.
This one?
11. No, 10. 10, the previous one. Yeah. What is the difference between the two calculated values in the third and fourth column in the table?
Yeah, because this is done experimentally here. Okay, so the first column is
no. Why are the two columns for calculation? Why are the two columns for experiment?
Yeah, so the first column is for the drug. So 21.4 is for the ibuprofen. Okay.
And the carrier is second one. Okay.
Carrier is the second one. So here's a experiment for the drug, and here's a experiment for the carrier. And the difference between these two columns are here. Difference between the experiment columns are in the second last column.
Yeah, thank you very much. There was one more question from the audience that just came, and the question is there a model for simulating drug release from liposomes?
Come again? What model?
Is there a model for simulating drug release from liposomes?
Yep, as I showed you, currently our scientists are working on that kind of examples, liposome examples. Again, this is in the time scale, it is kind of a longer time scale problem. Okay, so we are looking into that one. Yes, we are working on developing examples in that front, but it is ongoing work. We don't have a ready-made tool that is kind of take and apply or a plug in and play. So we don't have a tool like that. We have to build the models from the scratch. If you are ready to spend time and build a model from scratch, yes, definitely there is a way to do that particular liposome drug release understanding also.
That's it from me, Sylvia.
Thanks, Dr. Sudarshan and Dr. Kaushik for that question. I think I have covered all the questions that we have received. And I will just move forward to some of the questions that we have received about the licensing and the download part. So I would like to just quickly show you how to download the software. So can you see my screen?
Yes.
Thank you. So if you go to Schrodinger.com, this is how it looks, right? So on top, if you see this small download icon, this is where you have to go. If you click here, it will take you to this page. What you have to do here is you need to select, so this is the latest version that we have. You have to select what kind of OS you have. Make sure that you uncheck this button because you don't need this, and then just check this button and click on download, so you will be able to download it. So I went to this page because I already have a Schrodinger account. If you do not have it, it will ask you, or if you're not logged in, then it will ask you to log in. And if you do not have a website account, it will also ask you to register. So since that was not prompted for me, I will just go to that page. So if you do not have Schrodinger account, you have to go to Schrodinger.com and request hyphen account. Okay. So when you go here, you will see this little form. Make sure you fill this up according to your understanding. It doesn't have to be perfect. Here, you need to fill all the necessary information that is required and then submit. So when you submit, you will receive an email to verify your email. So once you click on the link to verify your email, it will go to our help team, which is our support team. So they will look over this information and then send it to the regional account manager, right? So the regional account manager will manually check it again and confirm this, right? After that, it will be activated. So because that is why there is little bit of a delay, as a lot of you asked. So sometimes what happens is we receive a lot of website account requests like now, because all of you are trying to register and trying to get this account. So there could be a delay. We usually do approve it within 48 hours. That we say, even when you click, you will receive that message that it will be activated in 48 hours. But sometimes it takes longer than that, and we are trying to make it as fast as possible. If you really are in a hurry, if something is urgent, you can write to help@schrodinger.com and they will quickly approve your website account. So that's it. The other questions, I think I will just show my slide once. So here, if you see, we have, so these are two registration pages. So you have to go to request account to get the website account. And if you have to register to this course, you have to go to DD course. Even if you have registered to this course, you will still need to go and request a website account. As I said last time also, there are two forms that you need to fill. I mean, there are more than two, but for now there are two. First, you have to go and register to this course if you haven't done it. So this is where you go, www.schrodinger.com/DD course. You will find a link to register to this course here. So please fill that out. The next thing that you have to do is to request an account on Schrodinger website. For that, you need to go to request account. So that is www.schrodinger.com/request-account, which I just showed you, right? So you have to have the website account in order to download the software and also to register for the Schrodinger online course. So I have a new update on the Schrodinger online course, Introduction to Molecular Modeling. Some of you asked, are interested and keen to register. So the next session is on November 2nd, and the registration and deadline and registration and payment deadline is October 26th. So we were able to get a discounted price, which is $125. The discount coupon will be sent to you via email. It was supposed to go out today, but I think it was delayed and will be sent out to you tomorrow morning. So please check your emails tomorrow morning and you will receive the discount coupon if you're interested in registering for this course. Other than that, we have office hours today. So our office hours are always on every Thursday, 4:30 p.m. to 5:30 p.m. So if you want to attend these office hours, if you have some questions, you can come and ask us. We'll all be there. You can get the link to join these sessions on this page. So if you go to www.schrodinger.com/dd course, you will be able to see the Zoom link and the password to attend these sessions. So the other thing is tomorrow we have no sessions. Tomorrow being the national holiday in India, we do not have any sessions, but we do have a session on Saturday at 10:30 a.m. This session is by our industry partner, Syngene International. We also have many exciting talks by our industry partner coming up next week and after. To check the new updated schedule, you may just keep looking at this page, and we'll also be sending you emails of any other updates. Other than that, if you have any hardware related questions, you can write to us at ddcourse@schrodinger.com. And for any other general query, for the evaluation license, we will be sending out instructions for you. So if you have already downloaded and installed your software, do not worry about it, but you will have to send us information, one file that we require in order to get you the license. So once your evaluation is approved, we will be sending out an email to you, and you will just have to send us that information, and we will try to get you the license within 24 hours. That's it from my end. Did I miss anything, my panelists?
No, that covers all.
Can you
Right.
mention the fee for the associate again? Some people might have missed it. They'll get an email for sure.
Yeah, so you will be getting an email tomorrow morning. So please keep an eye on that. The cost is $125 for all of you. There will be a coupon, a code will be sent to you.
What is the actual price and how much is the discounted price?
So the actual price is $350. This is a student price, and if you are student or non-student, we are able to offer it to you at $125. Otherwise, it is $350 for students and $475, if I'm not wrong, for non-students. But you can register and avail $125 if you are a student or not. Yeah. Anything else that I missed?
The other question is, if a person registered for this webinar, do they also have to have registered separately for the certificate?
No, that is not required. So if you're already registered, we will be sending you the emails to how to receive a certificate. So you don't have to register for anything else for now.
So if I install the software, how to get the license?
Yes, I think I already went over that. So when you have installed the software, wait for our email. My colleague Kishore will be writing to you and will be sending you instructions on how to send us the details in order to get you the license. So once you have received that email, you can send us the information and we will send you the license within 24 hours.
From Monday onwards, we will be having hands-on demo sessions.
Yeah, so exciting news. Anything else?
Will the course content be available after the completion of the course?
I think you will be a better suited person to answer this question, I think.
Yes, it will be there in JSS University YouTube page and Manipal University, their Manipal Schrodinger Simulation Center page. So it will be there for some time.
Oh, I thought they were asking for SOC. So I just wanted to check with you, Kaushik, are we able to keep some of the PDFs and some of the handouts there when you go through the SOC? Is there any content that people get to save?
Yeah, the SOC one, you can download all the content that is available over there. They have so many links that you can access to. Yes, you can access all of the content over there, and you can save them locally.
Thank you. So I think that's it from us. Thank you for joining today. Again, as a reminder, there is no session tomorrow, but there is a session on Saturday. Please join us at 10:30 a.m. on Saturday by a talk by Dr. Achintya Saha, or Das, sorry, Dr. Achintya Das from Syngene International. So have a great day, and we'll see you next on Saturday. Bye-bye.